So thank you for the introduction. I'm very glad that Dr. Lehman's video was shown. That makes my job a little bit easier. As was mentioned, I'm in the 4th year of my PhD. I specialize in biochar characterization. So today you're not going to hear from me a lot about field work or a lot about how to make the best biochar. My job is to describe what biochar looks like, how the different kinds of char are different, and how we can study them. Okay, we'll do this way. Today I'm going to talk about a couple things. First, I have to explain about the organization I work for, so a little bit about that. I'll talk about the 3 thermochemical processes we do at CSET, the overall goal of biochar engineering, 3 studies I've undertaken, and some of my future plans. So at Iowa State University, we have the Center for Sustainable Environmental Technologies, or CSET, and we're very proud to show off our brand new building. We just moved into it this summer. It's called the Biorenewables Research Laboratory, and it hosts several organizations. The first is CSET, which I work for, as well as a National Science Foundation Research Center, CBERC, our Bio-Based Industry Center, and an overarching organization called the Bioeconomy Institute. What does CSET do? Well, anything that involves thermochemical or biomass is probably in our realm of what we'd be interested in. So we do feedstock handling, pre-processing, our 2 processes, fast pyrolysis and gasification, characterization of all of our products, some upgrading of certain products for different applications, and finally some calculations of the techno-economics. We work at several different scales. For our fast pyrolyzers, we have 3 main scales. There's the ones you see here, which are a very small scale. This is done for reaction mechanism work, so a couple milligrams at a time. We have 3 bench scale reactors, so 100 grams to a kilogram at a time. So it works on the very small scale. We move up to the bench scale. And finally, we move it out to our process development unit at our BioCentury research farm. So this is an image of our new reactor room, still being put together. We have the 3 reactors in here. One is this free-fall drop tube reactor right here. It's very simple. You essentially put the biomass in at the top. It falls down through a heated column and comes out the bottom. We've got a fluidized bed. This is the technology that we've focused on the most. And then an auger reactor that will be constructed back here. And that's simply twin screws rotating to move the biomass along. What processes do we do? Well, pyrolysis first. Pyrolysis in general is heating without oxygen, as Dr. Lehman was so kind to mention in his video. And 2 types, slow and fast. And the slow and fast refers to the heating rate. How quickly do you heat up the biomass, and how long is it in the reactor? So slow pyrolysis would have residence times of minutes to hours, sometimes even to days. Fast pyrolysis, as it implies, is very fast. We heat it up very quickly, approximately 1,000 degrees C per second. And we try to get it in and out of the reactor as quickly as possible, about 2 seconds in the reactor. Our temperature range is fairly moderate. The primary product from slow pyrolysis is char, the solid. And from biomass, we're aiming for this liquid bio-oil product. Yields, you can get about 1/3 on average with slow pyrolysis, all depending on your conditions. We average about 12 to 15% in our fast pyrolyzers. We also do gasification. This is similar to pyrolysis, but as mentioned here, it uses some oxygen, so about 1/5 to 1/4 of what you would need for combustion. And that produces mostly a syngas at these higher temperatures. And that gets somewhere under 15% of char. So you make char in all these processes, but each char is going to be slightly different, and you'll get a different amount each time. I'm going to kind of skip through these slides, as Dr. Lehman did a wonderful job of introducing them. So this is what is biochar. You see the logo for IBI, and also the fact that it's got a long history and can make energy carbon negative. Several effects on soil. Again, he covered this pretty well. These are some of them. Nutrient availability, microbial activity, organic matter, decreases greenhouse gas emissions, fertilizer needs, et cetera, et cetera. So what's the idea behind biochar engineering? This is where I actually became interested as a chemical engineer. I saw that we were making all these different kinds of char, and they would just sit by the reactor once the reaction was done. And I wanted to know how these relate to the chars that Dr. Lehman was talking about. So we talked and came up with this kind of cyclical scheme of how you would engineer a char for a specific purpose. So it starts with what I specialize in is characterization. Before you can do anything with your char, you need to know what kind of char you made. And then, with the help of agronomists and soil scientists and plant scientists, we figure out how well does our char work. Once you know what it's like and you know how it works, you can guess, well, then what makes a good biochar for this situation? And once you know what makes a good char, then you can try to produce it. And if you repeat this cycle over and over again, the idea is you'll find the ideal char for a given situation. So the 3 studies I talked about, the first one was an initial characterization study. And the idea of this study was, what techniques do I even use to characterize biochar? So you start from the literature, you look at what they do for activated carbons, you look at what they do for charcoal fuels, and we came up with a few methods we can use. We used 7 chars. We specialize in switchgrass and corn stover handling, so we put those through all 3 of our processes, and we got a commercial hardwood char for comparison. And some things I want to point out from the study is that we found that switchgrass and corn stover have really high ash contents compared to wood. It's one of the key differences between them. That ash also, or the carbon is fairly low in nitrogen, so they have high carbon to nitrogen ratios, and our surface areas are fairly low. This kind of surprised me. You see activated carbons have surface areas in the hundreds, and ours were 1, 2, 10, much smaller. One key difference between the chars that come out of every process is the size. And this is something you notice right away if you see our processes. We put this switchgrass feedstock into all 3 of our kinds of reactors, and you can see the products that come out. So the same size went in. When you get out slow pyrolysis char, it tends to look exactly like the feedstock you put in. It's just black and it's more porous. You put it through a fast pyrolyzer, and those high heating rates tend to make the particles fracture into tiny little pieces. So this char is going to be a lot finer. And gasification char is somewhere in the middle. That's one way I can eyeball it, and if you show me what the feedstock looked like, I can make a pretty good guess of what process it went through. Some things about the inorganic composition. There's a lot of information up here, but I want you to focus on 2 things. The first is that if you look at a hardwood char, the main component of the minerals is calcium. Okay, and that's what you'd expect to find, alkali earth metal. But if you look at switchgrass and corn stover, it's not calcium, it's silica. And that'll make a big difference in its properties on soil. Silica is not basic like calcium is, and so it's not going to have the liming capacity, and it may also prove challenging when handling it. Because what's silica the main component of? Glass. And if you put lots of silica in a high temperature reactor, you will have made glass. And so this may prove a challenge when processing it, and you have to keep in mind this is not going to process like your wood chars. What we found from this initial study is that one technique that's very useful in characterizing char from a scientific view is solid state NMR. NMR standing for nuclear magnetic resonance spectroscopy. Very detailed technique. You're not going to find these in most labs, but they're very useful. And the reason they're useful is that they can tell you not only what kinds of carbon are present in char, but how much, which can be very hard to do from other techniques. And so these are some of our switchgrass chars. And you can see these are spectra. Almost all the spectra are dominated by these aromatic peaks. Aromatic carbon is what we're looking for. But there's a few other smaller things. But these, with the techniques that we use, will actually be representative of the types of carbon that are present. And we can analyze those spectra using standards and come up with actually how much is in there, given percentages. So you can see how many are carbonyl carbons. So these would be your carboxyl groups. How many of these aromatic rings, some that are non-protonated, so they're not touching a hydrogen, and some that are, and that'll be very important later, and then the different alkyl groups. So we can come up with that kind of information, which as an engineer is very useful. If I don't have a number to work with, I get very upset. I like having numbers. So this technique allows me to have numbers. What it can also tell us, using a different technique called dephasing, is how big these rings are. Because one of the debates in the biochar field, at least on the characterization side, is what does this stuff look like chemically? It's a huge mixture of different kinds of carbon compounds, but we wanted to get an idea of if you made a char molecule, and if there was a single average molecule, what would it look like? And that's what this technique allows us to do. So what we did is we put samples standard samples in that we knew how far the hydrogen was away from the carbon. In NMR, the protons interact with the carbon and make their signals disappear. So you can see here, signal there, over time it disappears. And the closer the carbon is to the hydrogen, the faster the signal disappears. So we know what 2 carbons away looks like, the blue line. We know what 3 carbons away look like, that's the red line. And then we plot our chars. And you can see with the green squares, we can guess that on average it's about 3 carbons away. And then our other char, char B, is going to be a little further away. We're still looking for good standards to get for the bigger molecules. And from all the information, from the quantitative information of what kind of functional groups we have, as well as how far away carbons are from hydrogens, we can make these kind of representative molecules. And you'll notice that a fast pyrolysis char and slow pyrolysis char are very similar. And we think that's because they're done at similar temperatures, and that gasification chars are much bigger. They're run at higher temperatures. So our thought from that study was temperature makes a big difference. Moving on to the next study. My colleague, Rachel Unger from the agronomy department, wanted to do a small field plot study. But we needed to know which chars to make because we'd have to make them in large quantities for this. Even for a microplot, you have to make quite a bit. And rather than make 17 different chars, we thought, well, let's just do a small study with 17 different chars that we have and use that to pick out the best ones. So you can see us there with our approximately 580 different soil incubation jars. Took a long time. 17 different chars, and we knew all of their conditions. That was the one requirement for participating in this study. It had to be made out of the 3 feedstocks we were looking at, and we had to know the reaction conditions. There was a lot of data from this study. I'm only going to show you 2 points. Otherwise, most of you would probably get dizzy and fall asleep. It's quite a lot of data. The first is after incubating for 8 weeks, we checked the pH. And here's the control soil, so that had no char added. We played around with adding urea or not adding urea. One thing you'll notice is that in general the pH goes up. This has to do with the basic ash content. Char has a small amount of liming capacity, especially when it's made out of these high ash feedstocks. That's one thing we saw very easily, and different chars would have different effects. We looked at cation exchange capacity. This is soil's ability to hold on to positively charged nutrients, and it didn't change much. We were a little concerned about that because we've seen in some literature that biochar increases cation exchange capacity, but then we thought, oh yeah, we only did this for 8 weeks. That may not be enough time for it to develop those reactive groups. So that's from the 17 char study. But what was most interesting out of the 17 char study is we noticed 3 of the chars did something special. They were made out of corn stover. One was made in our fluidized bed pyrolyzer, normal conditions, 500 degrees, very normal char. And 2 of them were made on a free-fall pyrolyzer while it was being set up. And these particular chars didn't get the appropriate amount of heat transfer, and so while the reactor said 600 degrees, they didn't actually achieve 600 degrees, and they came out kind of brown, which was fascinating to me to study. I thought, well, this will be a good char to include in the study, just because you can represent what it'd be like to have a really low temperature char. Also, when we ran the NMR, we saw this. Here's what corn stover looks like by NMR. Most of these peaks are the sugar molecules, and there's not much aromatic carbon. This one achieved the lowest temperature. This was kind of a medium brown. And you can still see some of the corn stover peaks, but you start to see the aromatic show up. You get a little darker color brown, slightly higher temperature, even more so. And finally, a typical char down there dominated by an aromatic peak. And I thought this is a great char to study because we can track the amount of reaction that's gone on. So I thought, well, let's take a look at bioavailability. How does the amount of time it spends in the reactor at what temperature affect how bioavailable it is? Now you're asking, what does bioavailable mean? Well, in short, it means able to be metabolized. It can be food. Specifically in this case, metabolized to carbon dioxide and by microorganisms. That's what I mean by bioavailable. Why does bioavailable matter? Well, Dr. Steiner, who you saw in the video, presented this study last year in Colorado, and he showed that how much carbon is available to microorganisms affects the growth of plants. The reason is, if you have soil microorganisms, if they can't eat any carbon, they will eat it. But they don't just need carbon. Microorganisms do not live on bread alone, shall we say. They also need nitrogen. And so if they have carbon to eat but not enough nitrogen, they will scavenge all the nitrogen from the soil around them, which leaves very little nitrogen for the plants. So in this study, there was some carbonized corn stover, so it's char, and there was some regular corn stover, and they're next to each other. Here's just a little bit. You don't see much difference, but you get to a lot more corn stover, which has more bioavailable carbon, and you can see what's called nitrogen immobilization. That nitrogen is no longer available for the plants, and the plants suffer for it. And so the idea is we want to have the right amount of bioavailable carbon because we don't want our microbes taking away our plants' nitrogen. The other thing, if you're on a global carbon cycle, bioavailable carbon, if you can eat it, it's not sequestered carbon. And one thing we want to do with biochar is sequester carbon. So bioavailable also not so good over there. So we looked at these 3 chars. We analyzed some corn stover first, and then put these in order of, I'm going to call it degree of pyrolysis. How pyrolyzed were they? And you'll notice several things across the board. Your volatiles go down as you pyrolyze it. Your fixed carbon, it goes up to a point. Your ash increases, because with ash, it's always concentrated in the solid. So as you pyrolyze it further, that gets more and more concentrated. So that's where the ash goes up. Carbon goes slightly down as you react it further. More of it's going to volatilize off. But what I really want you to focus on are these values down here. In the biochar community, we're trying to come up with standards of what makes a good char and how to measure this amount of bioavailability. And there's been several ratios or indices that have been proposed of how do you measure this. And one of them is a hydrogen to carbon molar ratio, how much hydrogen is there to how much carbon. And you can see it decreases over the degree of pyrolysis. So this may be a good measure. Some other people have looked at oxygen to carbon, also decreases. Or they've looked at C to N ratio if you want to directly measure how much carbon there is to how much nitrogen you have. Some people have looked at so-called fixed carbon versus volatile carbon, and see that increases as you have more fixed carbon and less volatiles. And in the NMR field, we like aromaticity. How many of those carbons are now aromatic versus aliphatic that they were before? And that increases. So lots of things you can use to measure. Those 2 pieces of information, the hydrogen to carbon and the oxygen to carbon, are often plotted this way. So it's called a Van Krevelen plot. And I've got some data from literature. This was one paper on chars made by slow pyrolysis at different temperatures, and this to me made a beautiful figure because you can watch it go from pyrolyzed all the way through the different temperatures down. I like this very much. And then we had some other ones. This group right here was done at a torrefaction study. Torrefaction is essentially low temperature pyrolysis. You're more trying to drive the water and the volatiles off. So you'll do a low temperature, and those are about 200 to 300 degrees. So you see they all fall in the same range. And then there was one other study that was done with red pine, and you can see its temperature range there. And you notice it looks different than the others. And I hypothesize that one of those reasons was that this reactor had some oxygen that was able to leak into it. I think that had an effect on the ending ratio. But I thought, well, if I don't know what temperature my chars actually made it to, maybe if I compare them to this line, I can make a good guess. So here's Stover in that yellow square up there. Biochar 1 appears somewhere around 350. Biochar 2 somewhere about 375 or 400. And then finally this one about 450. So that can be used one way of estimating what temperature your char reached. Okay, so that's enough about the char characteristics. Now let's give me some soil data. We put our chars in a loamy fine sand. The idea is we didn't want too much organic matter already there. We wanted something fairly sandy. We added about 0.5 weight%, which is pretty typical for a soil study. We added some nutrient content just to make sure the nutrients weren't limiting. Incubated it in the inside of a refrigerator for about 24 weeks at room temperature. We took some soil samples at 8 weeks to measure soil nutrients, did 9 replicates apiece, and we measured the CO2 coming off by an alkaline trap. It's hard to see in this picture, but inside all of those jars are little tubes full of sodium hydroxide that will trap the CO2 as it's given off, and then we can measure how much CO2 is being emitted. Some data. As expected, the materials that seem to have the most bioavailable carbon gave off the most CO2 because the microorganisms were able to eat them and give off CO2. So here's green is corn stover, and you can see most of the activity happened in about the first 4 or 6 weeks. And the chars at least at the beginning of the study, went in the order I was hoping they would. The more bioavailable carbon they had, according to all of those ratios I talked about, the more CO2 they give off, and so the more bioavailable they actually appeared. But even by the end of the study, you can see most of the chars were fairly similar to the control. So most of that bioavailable carbon has already been eaten. Plotted a different way, this has to do with the carbon sequestration aspect. If you look at Stover, by the end of 8 weeks when we did the first set of soil sampling, you can see that almost half of the carbon that was in the biomass is gone. It's been eaten. It was that bioavailable. And so that's not going to stay around for very long in the soil as an amendment. But if you look at the chars, even our 2 brown chars only lost about 10%. And our perfectly black char lost less than 5%. So you can see the effect that pyrolysis has on char stability. I do have to point out, though, this is a short-term study. This is not going to tell you what's going to happen over decades, just what happens shortly. Again, I'm an engineer. I apologize. I had to make it into a model and graph it. And this talks about the rate of CO2 evolution. So with Stover, you can see the rate is very fast at the beginning and then kind of evens out. With the 2 chars, these are the 2 brown chars, the 2 incompletely pyrolyzed ones. You notice that the rate as the slope here are very similar. This kind of surprised me because I expected them to kind of move evenly down. Based on the ratios we looked at, you'd expect biochar 2 to be about halfway in between, and instead it looked very similar, whereas char 1 had about half the rate. So that was a bit of a surprise. I'm going to kind of skip through the soil properties. I just wanted to throw these up there for the soil scientists in the audience. Big change we saw was pH. Again, the ash content in the chars. Slight change in a few other things. Potassium. About the bioavailable nitrogen, there's 2 forms of nitrogen that plants like the most, nitrate and ammonium. And you can see with the Stover, it's lower in both of those than the control. That's the carbon, or that's the nitrogen mobilization I was talking about. You can see the microorganisms taking the plant available carbon. Okay. One organization that's interested in char for a completely different reason is the UN Council on Prevention of Desertification. We've seen in some research that char increases water retention capacity, and so we decided to measure that here. And it's measured in terms of tension pressure. This is how much pull the plant has to exert to get the water out of the soil. The higher the tension, the drier the soil. And so this represents about field capacity. You can see at field capacity, there's a slight difference between all the amended soils and the control, but it's not statistically significant. And at higher pressure, this is where it really matters. When the soil is dry, how much can it hold on better than the control? And you can see the Stover helped a little bit. A couple of the chars did, but again, statistically significant, it was not. So more researches need to be done in that area to see how char helps with water retention. Had to study the microbial populations, and what you'll see here is that the more bioavailable carbon there is, the more food, the more microbes. So the chars and the control were very similar, but that Stover, where we added all that bioavailable carbon, the populations seriously spiked. Okay, so from that graph, those of you interested in references, here they are, right quick. So some conclusions from all 3 of these studies. What I want you to walk away with from today is that fast pyrolysis and gasification chars are physically and chemically different than your traditional slow pyrolysis hardwood chars for multiple reasons, some of which you saw. For that reason, these chars should be included in biochar trials. They are a kind of biochar, and we need to figure out what kind of char is best for the soil. So we should include those in all of our studies. The types of carbon present in the chars, mostly by NMR, seems to be very dependent on temperature and to some extent reaction time. This is the concept I'm going to base my final dissertation on, is that study of temperature relations. Co-production of biochar and bioenergy may be more cost-effective and resource-efficient for these biomass crops, especially those with high silica. Corn stovers and switchgrasses may not be the type of thing you want to use for traditional charcoal because of the silica, because of the high ash content. And biochar may be a solution to that. It might be a use for these feedstocks that is not going to be detrimental to your equipment. The soil does not mind if you put a little more silica on it. Your high-temperature combustor, however, may care. And finally, advanced NMR, the techniques that I showed you, may provide some of the best information for engineering char on the scientific level because it can provide the quantitative information that we need. So with that, future work, the microbiology study, the bioavailable study is almost ready. I'm hoping very soon to be submitted to soil biology and biochemistry. We're going to be looking at the temperature relationships, as mentioned before. We're working on developing NMR methods to track char oxidation. And that's because the char out of the reactor is very different than we see in terra preta soils. And we believe all of the changes are due to oxidation both from abiotic, so non-living processes, and biotic processes. But we don't have a good way of tracking that. We want to know how quickly that char changes. And so we're trying to develop NMR methods to do that. And finally, safety testing of fast pyrolysis chars. This will come up in some other people's presentations, I'm sure, today. We have two big problems. Remember the size of the fast pyrolysis char? Makes it very dusty. It can be very difficult to handle, and that's an issue that we have to address. Secondly, as with any charcoal material, it has this bad habit of, on very certain occasions, self-combusting. And we need to figure out how to prevent it from doing that. It is a very flammable material, and if you get it in the right conditions and don't handle it properly, that is a concern. So we need to figure out especially how do we handle these different kinds of chars. A lot of people to acknowledge. Funding from our Plant Sciences Institute on campus, as well as the NSF for my work. My major advisor, Robert Brown, as well as David Laird. Dr. Schmidt-Rohr has been teaching me about the NMR and helping me develop these techniques. My colleagues at CSET for making all of the chars. My colleague in agronomy, several colleagues in agronomy for various methods. The graduate students help with NMR, and people with the SEM, and finally 2 undergraduates who gave their assistance over summers to help me with these projects. But before I go for questions, I have to make a shameless plug for a conference we're going to be hosting in 2 weeks, the Symposium on Thermal and Catalytic Sciences. This will be people from industry, government, and academia, in many cases from around the world, specializing in fast pyrolysis, gasification, and all of the products associated with those processes. So with that, I thank you for attention and would be happy to answer questions.
Could you repeat the question so that we get it out of it?
From an energy perspective, has any thought been looking at the different energy use in producing these different types of chars, and then in the energy in the byproducts of the char, the gas and the oil?
Okay, so if I understand your question, I have to repeat this for the video. You want to know about the energy balances associated with all these processes. And that is something we've looked at very closely and that you'll hear more about from the people who do more gasification or people who do more fast pyrolysis. In all cases, you want to use as many of the products as possible and as much as the energy as possible. You want as many closed energy loops. In some cases, the energy will be a valuable product in itself. As you saw with the farmer with the gasification, he needed that for heating. In other processes, pyrolysis itself is slightly endothermic. You need to put energy in to make it go in most cases. And so some of the energy from your process is going to have to go to keep the process going itself. With the idea of fast pyrolysis, you want most of your energy to go into your oil because that's your primary product. Your syngas will provide the energy to run the process, so that'll be combusted on site. And then the char would not be used for energy, it'd be sent out to the field instead. One thing that I've been trying very hard to do is to get other engineers to think about putting the char in the soil rather than combusting it, because they're concerned about their energy balance. The char is an energy product in most cases. With gasification, the majority of our energy goes as a syngas, and the char is simply left over. But gasification, as a partial combustion process, tends to be more exothermic, so it can provide its own energy. But pyrolysis, you have to recycle some of it back in. Does that?
Pyrolysis is energy self-sufficient, though it's slightly endothermic. What happens is that the products has also energy, has energy content that you can, like you saw the video, you could use the products to run the process. Right, so essentially, it's energy self-sufficient.
An ideal pyrolyzer would require no external energy in. All the energy would either come from a product of the process or additional biomass that you're using. But ideally, you would have a energy self-sufficient process that produces energy products.
Essentially, if you want a closure, you could say about 80%, 85% closure. So you have essentially it's a furnace, so you have heat losses.
The solid-state NMR is really interesting. Do you do any wet chemistry to confirm that for carboxyl and phenol?
The question was, do we use any wet chemistry to supplement the NMR data? At this point, we haven't. We do know of things like a Boehm titration. I always mispronounce that, forgive me. And that'll help quantify by wet chemistry method how many carboxyl groups there are versus how many phenolic groups, et cetera. We know of that method. We haven't tried it yet. We found with NMR it to be very consistent between samples and very reproducible, but it will be something that needs to be verified in the future. But so far with this technique, one reason we've stuck with it, it is very reproducible and very accurate.
Well, this question is related to the first question. Is there any way to evaluate the ratio between the carbon footprint for the production process versus the sequestered carbon from biochar?
The question was, I'm guessing your question has to do with life cycle analyses of how do you relate the carbon footprint of the process to the carbon sequestration. I haven't done that kind of work myself. Several people in my group have looked at, as part of their their techno-economic analysis of how much carbon you have to use in the process to supply your energy. So how much carbon goes towards heat or towards other energy versus how much carbon ends up in the char that you can sequester. And I think in general that'll be a very delicate balance because you're going to need some amount of carbon to create the energy products that you're going to sell to support your process, and that'll have to balance with how much carbon you want to sequester. So there's always going to be this act. The carbon has to go to each of those places, and you have to supply both. And so figuring out how that balance is going to work will depend on your situation. Does that help?
I think you presented it, and I missed it. If you talk about carbon-nitrogen ratio, is there kind of a sweet spot that you're seeking in the char for carbon-nitrogen ratio?
The question was about the carbon to nitrogen ratio. And with char, that's tricky. Because with plant matter, generally all of the carbon that's in it is bioavailable. And with carbon to nitrogen, it doesn't really matter what the total C to N is, it's the total bioavailable C to the total bioavailable N. And with microbes in soil, you want to aim somewhere around 20 to 30. And soil microbiologists in the room, please correct me if I'm slightly off. And that's just enough nitrogen that the microbes can have all the energy that they need, the nitrogen that they need, and there's still nitrogen left over for the plant. If you have a ratio higher than that, you'll start running into immobilization problems.
You were mentioning about the bioavailability of the biochars, or the carbon from the biochars. So if I understand you correctly, if you put maybe a kilogram of biochar in the soil, you're not actually sequestering a kilogram or the carbon equivalent of 1 kilogram of biochar because some of that carbon is bioavailable. So even though you might put some in the soil, some percentage of that is actually going to the atmosphere. Is that right?
The question was, if I put down a certain amount of carbon in a kilogram of biochar, how much of that is actually sequestered? And that is a very good question. It will depend very much on the char properties. One thing we want to look at, because all carbon in soil eventually has to return as CO2. Otherwise, we'd be waiting in pools of charcoal. But it's a matter of how quickly does it go away. And we're seeing a relationship between the types of carbon that are in a biochar and how much of that will be bioavailable on the short term. So you saw from my study, in a very crude way, that most biochar has unavailable carbon. Most of the carbon in char is not going anywhere anytime soon, but there's a tiny fraction. Most studies have a couple percent within a short period of time, so within a year or two. The half-lives are mostly ranging on the hundreds of thousands of years. So you're going to lose some, but I would think it's a very safe bet. If you have a black biochar, so a nice, dark, properly pyrolyzed one, I would expect you're not going to lose more than a couple percent on the short term.
Also, Cody, I think your data shows that depends actually on the volatile content of the char. It's the biochar itself remaining in the biochar. You see the correlate very well with the volatile content. So you have biochar number 3, which is actually low volatile content. In fact, Macalantau has shown studies which the plant forage yield depends on the volatile matter remaining.
So the question was about the volatile matter. And you can see very clearly here, it decreases with pyrolysis, the degree of pyrolysis. The tricky thing with volatile matter is how it is measured versus how it's applied in soil. To measure volatile content, you essentially heat it up in the absence of oxygen and see how much mass is lost, how much can be volatilized by heat. And there seems to be a correlation between that and what microbes can eat, but how closely they're correlated, we've had trouble finding a very good pattern. I plotted actually volatile content versus the amount of carbon lost, what I called bioavailable carbon, and the line wasn't all that great. It does have a relationship, but it's not exact. And so what I think as a characterization community we need to define is a better measurement than volatiles. We need an actual measurement for what is bioavailable, not just what comes off with heat, because what comes off with heat is very different than what can be metabolized.
In your data, did you actually see any data point where, in the presence of high level of volatiles, the plant growth does not suffer? Are there any exceptions to what you normally expect?
We did not plant anything. Most of the people I've talked to only a couple people have seen these very drastic negative effects from high volatile content. There's only one study I can think of that was presented, I believe, 1.5 years ago. But in general, as long as you have properly pyrolyzed char, I really think that char didn't reach the temperature it needed to. And so the volatile content, even if it might have been similar, it was the type of carbon that was left, I think, caused more of the problem. And so that exact relationship, I'm not sure how that's going to work out. I think there is one, but again, I haven't done a lot with plants yet. Yes?
Did anybody look involved with the pyrolysis process as what their greenhouse gas potency is in comparison to what we're talking about here?
The vapors that come off of fast pyrolysis, so the volatiles, if you will, are what makes up bio-oil. And so the people who do bio-oil in the room can tell you all of the compounds that are in there. And ideally, you want to trap those volatiles as they come off. You saw the kilns where the smoke was just billowing out. The idea of fast pyrolysis is to catch those and make use of those compounds. Greenhouse gases themselves, they emit some CO2, a small amount of CO, but most of it's going to be a slew of water and organic compounds, many of which will come out of the atmosphere or hang up there more as particulate or aerosols. But it hasn't been studied as greenhouse gases themselves.
Yeah, I wonder, you are listing several surface areas.
Yes.
And those surface areas, they are less than 10 square meters per gram. And compared to activated carbon, you mentioned like hundreds or even thousands square meters per gram. So I want to say, is there any measures you can use to increase, or if you think you need a higher surface area for this application? We were able to get very high surface area even for coal under the pyrolysis condition up to 400 square meters per gram. From pyrolysis. So I assume we haven't done anything related to corn, but I assume corn even harder for pyrolysis and activation.
The question was concerned about our pathetically low surface areas from an activated carbon perspective. Activated carbon generally consists of two processes. Normally, you make the charcoal first, and then you put it through a second oxidation process, either high temperatures, steam, acid, et cetera. And Dr. Boateng can speak more directly to this. And it's that second process that reacts with carbons to create these tiny little pores, which is where most of the surface area comes from. Looking at our biochar, we have some pores, but most of them are the bigger pores. at least in the chars that we work with, which is one reason why you don't see very many corn stover activated carbons. Corn stover as a material is generally not so good for holding onto those micropore structures. Also, through the fast pyrolysis, as we make our particles fracture or explode, if you will, in the reactor, that it doesn't leave a lot of pores. Most of those pores have been exploded. And so you just have particles. Yes, I do not have the data on hand. But in general, you don't have a huge amount of micropore area. It tends towards zero. Most of it's in the meso and macropore, at least with these particular chars. So I don't think I would advocate using corn stover chars as an activated carbon to anyone if you base it solely on surface area and its ability to develop surface area. These chars are not for that.
Is a high surface area a desirable property? Or does it not matter at all as a soil amendment?
The question is, does surface area matter as a soil amendment? And I would say that is a really good question. We don't know.
I'll dwell on that a little bit later. Maybe leave some of these questions.
He's done a lot more work with the surface area side.
It depends. Yeah. If you're talking about climate exchange, it doesn't really matter.
Like biochar used in any kind of remediation, soil remediation?
The question was, is biochar used in soil remediation? And I would say there are a lot of people who are interested in just that. People are looking at reclaiming soils. They think that char, because of the absorptive properties that it have, may help in cleanup sites. Also, if it increases microbial activity, may help those processes along. So there are people looking into that. It's a great area of great interest. Was there a question in the back earlier?
I was wondering a little bit about the ash. What form is that, as the silica? Is it a discrete mineral, or is it maybe clogging up the pores? It seems like there's a lot of ash there, at least in the corn stove.
From what we can see using scanning electron micrographs, it seems to be in discrete particles rather than like a coating, but we haven't looked in great detail at the form of the silica there. Okay, and with that, I better hand it over to Dr. Spokas.